Welcome back to Studious. Today, we are putting human movement under the microscope. We are doing a complete structural teardown of skeletal muscle tissue, exploring everything from its heavy-duty macroscopic scaffolding down to its incredibly precise microscopic engines. When you lift a heavy weight, run a marathon, or simply maintain your posture against gravity, your body relies on a biological miracle of biomechanical engineering. Skeletal muscle is arguably the most highly organized tissue in the entire human body. It is designed for one singular violent purpose: to generate immense pulling force without tearing itself apart. To understand how we move, we have to look past the gross anatomy of the bicep or the quad and zoom all the way into the cellular level. In this video, we will decode the complex connective tissue wrappings, map the energy-packed sarcoplasm, and witness the sliding filament theory in action. Finally, we'll see what happens clinically when this flawless architecture breaks down. Let's dive in.
Before we focus on skeletal muscle, we must orient ourselves using a diagnostic matrix of the body's three distinct muscular engines. First, we have skeletal muscle, our primary focus today. It is entirely under voluntary control via the somatic nervous system, designed for high-force, directional pulling, and histologically characterized by heavily striated, highly organized parallel fibers. Second, we have cardiac muscle. This is an involuntary engine found strictly in the heart wall. It is designed for highly synchronized, continuous rhythmic pumping. Under a microscope, it is also striated, but its cells are uniquely branched and interconnected by specialized intercalated discs to share electrical signals instantly. Finally, we have smooth muscle. This is the body's slow, involuntary engine, lining our digestive tracts and blood vessels. It is built for regulatory compression and rhythmic squeezing. Histologically, it lacks the highly organized protein structure of the other two, giving it a smooth, completely non-striated appearance under the microscope.
To truly appreciate skeletal muscle histology, you have to know how to read the canvas. Throughout this video, we will be analyzing tissue primarily treated with the standard hematoxylin and eosin, or H&E, histological stain. This chemical combination is the gold standard in pathology. The eosin acts as the acidic dye. It strongly binds to the massive amounts of basic structural proteins packed inside the muscle cells. This is why the dense muscle cytoplasm stains a lush, vibrant, pinkish red color. The hematoxylin acts as the basic dye. It forcefully binds to acidic structures, specifically the DNA-rich chromatin inside the cell. Therefore, the nuclei will stain a dark, highly visible bluish purple, which is crucial for tracking cellular organization. To make this learning process as clear as possible, throughout our visual document, we will use a targeted digital green highlight to distinctly isolate and identify the specific microscopic structural components we are discussing. Let's start looking at the tissue.
Let's begin our teardown by looking at the macroscopic architecture. The best way to visualize skeletal muscle is by using the cable metaphor. An entire muscle organ, like your bicep, is not just one giant solid piece of meat. It is the outermost casing of a complex cable. This entire organ attaches directly to your skeletal bones via ultra-high tensile collagen tendons. If you slice that main muscle open, you will see that it is actually packed with smaller, grouped internal cables called fascicles, or muscle bundles. But we can go deeper. If you open up a single fascicle, you will find it is stuffed with dozens of individual hair-like strands known as myofibers, which are the actual individual muscle cells. So, biologically speaking, your muscles are essentially nested bags, bags within bags, full of these individual microscopic force-generating tubes perfectly aligned to pull in the exact same direction.
Because skeletal muscle generates such incredible, violent pulling force, it requires heavy-duty tensile scaffolding. If the muscle wasn't bound tightly together, it would literally rip itself apart during a heavy lift. This scaffolding consists of three distinct connective tissue layers. First, the epimysium. This is the tough outer layer made of dense, irregular connective tissue mixed with elastic fibers. It completely encircles the entire whole muscle organ, stretching and recoiling as you move. Moving inward, we hit the paramecium. This is the middle sheath that acts like biological cling film. It tightly wraps around the individual fascicles, ensuring those bundled groups of muscle cells fire and function as a unified structural unit. Finally, we have the delicate endomusium. This inner layer is made of loose connective tissue, featuring squiggly collagen fibers. It fills all the microscopic spaces between the individual myofibers, providing vital electrical insulation so an electrical signal sent to one cell doesn't accidentally trigger its neighbor.
These 3 layers of connective tissue also house the muscle's critical infrastructure support systems. Embedded deep within the endomusium are the builders, the fibroblasts. These are highly active cells that constantly manufacture and secrete the abundant, squiggly collagen fibers that give the delicate endomusium its physical integrity and resilience. But muscles also need external protection. Just outside the heavy epimysium, we find the shock absorbers, perimuscular fat. This layer is composed of massive clusters of adipocytes, or fat cells. They act as essential physical cushioning against blunt trauma and provide crucial thermal insulation to keep the highly active muscle warm. When you view this on an H&E slide, the fat cells look like large, empty white voids with tiny peripheral nuclei. This is because the harsh alcohol chemicals used during the slide preparation process completely dissolve and wash out the massive lipid droplets, leaving only the empty cellular outline behind.
Now we zoom in to level 1, the myocyte, the fundamental cellular unit of skeletal muscle, also referred to as the myofiber. These are not your average circular human cells. They are massive, elongated, perfectly cylindrical tubes that can run the entire length of a muscle. When you view them in a cross-sectional cut, you will notice a fascinating histological phenomenon regarding their nuclei. In a standard cell, the nucleus sits right in the center. But in a skeletal myocyte, you will see dozens of dark purple nuclei squeezed aggressively against the extreme outer edge, right against the cell membrane. Why? This is an incredible evolutionary adaptation. The interior of the myocyte is absolutely packed to the brim with sliding protein engines. By pushing the bulky nuclei to the extreme periphery, the cell ensures they do not physically obstruct or get crushed by the massive, violent mechanical pulling action happening deep inside the tube.
What actually exists inside this massive tubular cell? First, let's look at its outer boundary, the sarcolemma. This is not just a simple flat membrane. It features deep, microscopic, tube-like invaginations that dive straight into the core of the cell. This brilliant design allows incoming electrical action potentials from your nervous system to spread instantly into the deep interior, guaranteeing the entire massive cell contracts perfectly simultaneously. Inside the sarcolemma is the sarcoplasm, the muscle's specialized cytoplasm. Because contraction requires ridiculous amounts of energy, the sarcoplasm is heavily engineered. It houses massive localized concentrations of calcium ions, which are the chemical keys required to unlock the protein engines and trigger the physical contraction. It also contains glycosomes, which are massive granules of stored glycogen ready to be instantly burned for rapid ATP energy production, alongside specialized myoglobin proteins that hoard a localized reserve of oxygen for sustained aerobic activity.
If we look closely at the sarcoplasm, we reach zoom level 2. Suspended within the fluid are thousands of rod-like microscopic chains called myofibrils. These myofibrils run parallel to each other across the entire length of the cell, and they are the true physical engines of human movement. When you look at skeletal muscle under a microscope, its most defining visual characteristic is its transverse striations, the beautiful alternating pattern of light and dark bands stacked along the fiber. These striations are not just random visual patterns or cellular decorations. They are the direct, visible result of the highly precise, perfectly ordered, overlapping arrangement of thick and thin protein filaments densely packed inside these myofibrils. The exact geometric alignment of these protein filaments dictates how light passes through the tissue under a microscope, creating the distinct banded pattern that pathologists rely on to identify healthy skeletal and cardiac tissue.
Zooming into the ultimate micro level, we find the sarcomere. The long myofibril chain is divided into these tiny segmented contractile units. A sarcomere is physically bordered on each side by zigzagging protein boundaries called Z-lines. Between these Z-lines, we find the overlapping protein filaments. The dark bands, known as A bands, are composed of thick myosin proteins. The lighter bands, known as I bands, are composed of thin actin proteins. Muscular contraction is essentially just these two protein filaments sliding past one another. When a nerve triggers a contraction and calcium floods the area, the thick myosin heads reach up, physically grip the thin actin filaments, and forcefully pull them inward toward the center. Because the actin is physically attached to the outer Z-lines, this inward pulling drags the Z-lines closer together. This incredibly small, microscopic action physically shortens the entire sarcomere, converting chemical ATP energy directly into mechanical kinetic force.
To understand human motion, we must look at the synthesis, the chain of force. How does the microscopic sliding of two invisible proteins result in the swinging of a human arm? The answer is that force cascades upward through the architectural hierarchy. Step 1 is ignition. Deep inside the cell, myosin grips and pulls actin. Step 2 is microtension. The actin physically tugs on the Z-lines, shortening the microscopic sarcomere. Step 3 is cellular pull. Millions of sarcomeres shortening simultaneously shrink the entire myofibril chain, which forcefully pulls on the cell's outer sarcolemma. Step 4 is scaffold transfer. That cellular pulling force is transferred safely outward into the connective tissue wrappings, moving from the endomysium to the perimysium out to the thick epimysium. Step 5 is macroscopic action. The fully engaged muscle belly pulls directly on the high tensile collagen tendon, which forcefully acts on the rigid bone, physically moving your skeleton.
As future medical professionals, you must understand what happens when this perfect alignment fails. Let's look at muscular dystrophy, a genetic disorder defined by the catastrophic breakdown of this precise histological architecture. In a healthy anatomy, we see maximum cellular density, incredible structural uniformity, and those tightly constrained, perfectly aligned peripheral nuclei. However, in the pathology of muscular dystrophy, we see chaos. The H&E stain reveals highly disorganized, severely reduced myofibers. Massive cellular atrophy shrinks the muscle fibers, leaving immense empty void spaces filled with scar tissue. Crucially, look at the nuclei. The once rigidly organized peripheral nuclei become centrally located, wandering into the middle of the cell. This is a telltale diagnostic sign of structural collapse. This microscopic breakdown entirely severs the chain of force, leading to a tragic, progressive loss of macroscopic muscle mass, physical mobility, and eventually the ability to breathe. Thank you for studying with Studious. Please subscribe, like the video, and keep studying hard.